Thank you so much. Can you hear me clearly? Great. Thank you so much for inviting me. It's a pleasure to be here speaking to you all today. Okay, so you've been listening a lot about the cortex, and so for something completely different, we're going to be talking about white matter now. As Hiromasa explained really well in the introduction, white matter is actually 50% of the human brain. It's really important for synchronization of signals and communication between long-distance brain areas. More particularly, I'm going to be talking about plasticity in white matter. So when we think about plasticity, we actually usually think about neurons and synapses. And today I'm going to show you evidence that actually white matter, and more specifically, myelination, can be plastic and is important and plays a role in learning and acquisition of new skills. So for a very long time, we thought that white matter was stable throughout adulthood after development had finished, which in this case actually means, well, late into your 30s. But we didn't know that white matter could be changed with experience in learning. And in this seminal work by Heidi Johansenberg's lab, they demonstrated using diffusion tensor imaging, which you also heard a lot about today, that white matter could change. More specifically, they asked participants to learn to juggle and practice half an hour every day, and they scanned them before and after with diffusion tensor imaging. What they found was colocalized with increases in gray matter, which had been described before using the same exact motor model. They found increases in gray matter in areas of the brain that you see here in red that are related to eye-hand coordination, and colocalized with that, they found increases in fractional anisotropy that are described there in blue. So we know that diffusion tensor imaging is sensitive to things that are happening in the brain tissue, but we don't know exactly what is modulating it. And at the time, we had no evidence from animal studies that white matter was plastic, and so we didn't know exactly what was going on here. So we know that fractional anisotropy is modulated by these, here are just a few examples, by these things. For instance, if you have a lot of axons in a particular area, FA will tend to be higher there. If you have axons with large calibers or large diameters, FA will tend to be lower there. If you have areas of crossing fibers or disorganization of fibers, then FAs tend to be lower compared to areas where fibers are highly organized, like for instance the corpus callosum. And we also know from preclinical studies that myelination plays an important role in modulating these signals, and it's estimated in about 20% of modulating of FA values. Additionally, other things can be happening, like for instance, angiogenesis, which is just formation or pruning of blood vessels, and also astrocytic alterations. There can be also intracellular and extracellular space alteration that could modulate fractional anisotropy. So not everything is just purely cellular. But when we see these alterations in the juggling study, we actually don't know exactly what is corresponding to, because FA is sensitive to all these things. So there's not a one-to-one correspondence between an FA value and a particular feature of the tissue. So, as I said before, there was almost no evidence of white matter plasticity. There was some preclinical studies, and here quite preclinical, so they're in vitro slice or cell culture studies that had described that myelination is actually activity dependent. So when a neuron fires with high frequency, oligodendrocytes, which are the cells that make myelin, will tend to wrap more around these neurons compared to neurons that fire at lower frequency. So our main hypothesis for the potential cellular underpinnings for the things that we detected using DTI in the juggling study was potentially myelination. That's what we tested in the next study. So here we used a rodent model, where it's more specifically a rat, where we could train in a motor task and then also acquire the same technique that we acquired in humans and then perform a histological analysis. And again, here our hypothesis was more specific to myelination, so we actually didn't test for other things. So in this study, we actually had to perform it post-mortem, but speaking a bit more about the actual motor skill learning that we used, this is called a skill reaching task. I'm going to try to make the video work. It's a lot of motor stuff to try to, okay, so this rat is trained in a task called a skill reaching task. It has been previously used widely by behavioral neuroscientists to induce not only skill learning, but also to demonstrate that the motor cortex is plastic and there's reorganization of this area when these animals learn to reach and grasp a food pellet like you saw just there. As a control, we train rats to reach but not grasp the food pellet, and they don't develop these fine motor movements that are necessary to reach and grasp the food pellet, and that doesn't reorganize the motor cortex. And we also had a cage control. So here is important to have an active control because that was something that actually was missing from our human study in the juggling study, because we couldn't really convince people just to move their hands like this for half an hour every day. So we didn't have an active control. Here we have this active control that allows us to dissociate effects of performance, of learning the task, from effects of just increased use of the limb. And by design, our active control actually reaches more often, which is what you're seeing on graph B on the gray line, more often than the learning animal. So we can dissociate these two aspects. So the first question that we had was, can we even detect alterations in white matter using a rat model? As it was explained before, rats have very little white matter compared to humans, so it could be that even if we might not be able to detect anything first, they might actually not need white matter to perform this task, so white matter might not have an important role in the rodent brain as much as it does in the human brain. We were able actually to detect differences between groups. So we found that the learning group had higher fractional anisotropy in white matter tracts that were colocalized with sensory motor areas. Because these rats were also only trained on one paw, we actually only detected these alterations or these differences in the contralateral hemisphere, which controls the limb. We don't find, and that was compared to the passive control and to a passive control that doesn't do anything and the active control that just reaches but doesn't develop the fine motor skills. Additionally, we perform immunohistochemistry. Here, again, we had a hypothesis for myelin, so we stained it for myelin basic protein, which you just heard about as well, and we identified that only the learning group had higher myelin basic protein in the same areas as was identified with the DTI images. Additionally, the myelin basic protein was correlated with task performance in the contralateral hemisphere, but not in the ipsilateral hemisphere, and that's what the graphs that I'm showing at the bottom are exemplifying. Okay, so because we detected myelin basic protein alterations in these rats, next we actually performed this study again in vivo, and we use magnetization transfer ratio. This is a different MRI technique that is a bit more specific for myelin. It's not 100% specific for myelin as most MRI techniques won't be, but the things that we know is sensitive to are things like edema, inflammation, etc., which is important, for instance, for multiple sclerosis models. But because these are healthy adult rats, we weren't very concerned about those things like edema and inflammation. So we used it in vivo, and here we find, again, in this case, a change and not just a difference between groups. So an increase in MTR in the learning group, which you can see there in the red bar, in the contralateral hemisphere to the trained paw compared to the control group and the passive group. And this change in MTR also correlated with a measure of performance. However, not only complex motor skill learning, such as the skill reaching task, results in alterations in white matter. We also have been able to identify it by using other tasks, for instance, a complex sensory experience task. In this case, we use the DTI to guide not only the immunohistochemistry, but we also use it to perform mRNA expression analysis. And what we find is after these rats perform this task, there are alteration in fractional anisotropy in widespread areas of the white matter. There are now actually in slightly different tracts compared to the motor task, which is kind of expected. But then we also identified in these rats increases in myelin basic protein, again, compared to a passive control. And we also found there was an increase in myelin basic protein, but in this case, mRNA expression. It didn't survive multiple comparisons, but we found that there was a marker for oligodendrocyte precursor cells called PGFRA alpha that was downregulated in these rats. And I'm telling you this because this is important for future studies that I'm going to show you. And this potentially indicates that OPCs, oligodendrocyte precursor cells, that we know divide and are able to differentiate even in adulthood into mature oligodendrocytes, that they were downregulated, potentially indicating that they had differentiated and made new oligodendrocytes. Now, this is not just evidence from our lab. The other labs since the juggling study have started looking at these questions. In fact, one study using a combination of MRI and immunohistochemistry, like our approach from Yaniv Zasaf's lab, they found that cognitive learning, like spatial learning, more specifically the Morris-Water maze, induced also alterations in white matter and alterations in myelin basic protein. Additionally, using a different technique called two-photon microscopy, where you can track the same cell longitudinally in the upper layers of the cortex. They see that new oligodendrocytes are formed when the animals are exposed to new sensory enrichment. And neuronal activity alone can also induce rapid changes in oligodendrocytes and myelin. In this case, they used optogenetic stimulation for 20 minutes, and within 3 hours, they were able to detect an increase in new oligodendrocytes, so in oligodendrocyte precursor cells, and 24 hours later, they had differentiated into new oligodendrocytes. So this can happen quite quickly first, and also a number of learning and also experiences and neuronal activity alone can induce alterations in myelin and in white matter. Now, just to summarize what I just said, well, we detected alterations in white matter in humans. This seems to be related in part to myelination. And I want to say in part and highlight this because we did not investigate other mechanisms. So we know that in part there are alterations in myelination, but that's not the whole story. So there are likely other additional cellular mechanisms that are underlying also our changes in diffusion tensor imaging and other MRI metrics that we used. Now, focusing on myelination, and this is actually a really complex mechanism. As I was explaining before, there are cells called oligodendrocyte precursor cells, OPCs. These are the largest progenitor population in the central nervous system. They're about 5% of the total number of cells in the human brain. And I don't know if you can see that very clearly, but all those green dots that you're looking at in gray matter, in white matter, they're all OPCs. They're all over the place. And they remain active, they remain dynamic throughout adulthood, and they're able to differentiate, they're able to proliferate and differentiate into new oligodendrocytes. So potentially, when we're looking at an increase in myelination, these cells might be playing a role. Also, the pre-existing oligodendrocytes are already wrapping their myelin sheets around the axons. They can potentially also be playing a role by increasing the thickness or the length of the myelin sheet, as well as adjusting the length of nodes of Ranvier. Now, obviously, the opposite process can also occur, so thinning and shortening of the myelin sheets. and potentially even a retraction of myelin sheets. So when we see an increase in myelination, again, we don't know which one of these processes is happening. It's probably a combination of several. And another thing that I want to highlight about myelination is that we have this idea that areas like the corpus callosum or the optic nerve, that they're fully myelinated, and that's not true. You have unmyelinated axons in the middle, but even the same axon can have patches with myelin and other patches without myelin. So that's something to keep in mind, that what we see in the textbooks, that they're fully myelinated, really beautiful, that's actually not what occurs in the adult brain even. Okay, so we're interested in trying to understand where our increases in myelination could potentially be deriving from. And so we decided to test this by teaming up with Bill Richardson, that has a really nice knockout model where myelination, or the differentiation of myelination, can be knocked out conditionally in OPCs. What does this mean? This means that we can give a drug to activate the knockout at any age, and the oligodendrocyte precursor cells will be in the tissue, but they will be unable to become mature oligodendrocytes, so they can't make myelin, they can't put it on the circuit. And because we had this indication on the previous study that PGFRA, which is a marker for OPCs, was downregulated, this could potentially be one of the mechanisms that we were detecting. So Bill has really beautiful papers showing that when we knock out or inhibit this process in these cells, that there are behavioral effects. So these mice are unable to acquire a complex motor skill. Here, more specifically, was a complex wheel running. So some of the rungs are missing, and it's hard for the mice to run the wheel. But after a while, they get better and better and better. And you can see here that they're significantly different in their acquisition than their siblings that don't have this knockout. We first replicated these results in our lab, and the replication is actually really beautiful. It took us a while, but we managed to replicate the behavior. And the idea was that then we would scan these mice and see if we could detect differences using our MRI metrics. And this work was all done by Malte Kaller in the lab. Okay, so here again, we use magnetization transfer ratio because it's supposed to be a bit more myelin specific with the caveats that I mentioned before. And when we look at different regions of interest, for instance, like the corpus callosum or motor areas, we find a significant difference between the genotypes, indicating that MTR is actually sensitive to this very subtle manipulation that we're doing. You're just not allowing these cells to become mature oligodendrocytes. And so the interval between when we turn them off and when we scan them is about 2 to 3 weeks, sometimes a bit more, or actually 3 to 4 weeks. That's what's written there. So about a month. So there is already an effect in the microstructure that can be detected with MTR. So potentially, in part, these cells might have a contribution to the increases in myelination that we are detecting in previous studies, as well as pre-existing oligodendrocytes. And again, I'm not saying just myelin, so we know that other things might contribute to changes in our signal. These mice have also been used to demonstrate that when you, again, inhibit this particular process, this new myelination, these mice are also unable to perform as well in tasks like the Morris-Water maze. More specifically, their consolidation, so their acquisition of the Morris-Water maze is the same, but they are unable to remember it as well as compared to their siblings. All right, so beyond myelination. So I've been talking a lot about myelination because that's what we've been focusing on, but I want to again highlight that a lot of other things can alter diffusion tensor imaging signal, as well as MTR. So other things besides myelination can also be happening, like astrocyte swelling, axons diameter changing, and axon branching, for instance. besides myelination. And there is actually evidence from animal studies that all these things can happen with learning and experience right now. And here I'm just giving you an example because I've been talking about increases and decreases, but I haven't mentioned decreases. So not only positive experiences, learning or environmental enrichment, but also negative experience like sensory deprivation can also result in alterations in white matter, particularly here in decreases in myelin and also axon diameter reductions. This was not paired with MRI in this study, but you can imagine how this could potentially alter diffusion tensor imaging metrics compared to, for instance, MTR metrics, because you have a decrease in myelination, but also a reduction in axon diameter. You might actually not be able to detect anything on DTI because these two things might cancel each other out. But for instance, in MTR, you might be able to detect, if it's a bit more specific to myelination, you might be able to detect a decrease in the signal. All right, so to summarize then, what I want you to remember from today is that white matter is plastic, myelin is plastic. What we're detecting with MRI is not myelin specific. So we know that there are potentially additional cellular mechanisms that are happening at the same time that are modulating our signal and potentially in the same direction as myelin, and therefore we're detecting it. So most preclinical studies have only focused on myelin, but we haven't really looked at other things like diameter, et cetera, in combination with histology. So keep that in mind. Yes, myelin plasticity is happening, but we don't think that's what's modulating the entire MRI signal. And I would like to thank my team and the students that did a lot of the work, and also our funders, and to you for your attention. Thank you. And questions?
So now the talk is open to the question. If you have a question, please raise your hand or go to the microphone. It's free.
Thank you for the great talk. About white matter plasticity and plasticity in the brain in general, there is one aspect that really interests me is not with, well, it's also with acquiring new skills, but in a specific case, which is when you have a brain lesion that necessitates your brain to try to find new pathways and these kind of things. And I was wondering, have you tried, or have you the project of trying to see if you have a lesion somewhere in the brain? You can spot this kind of plasticity after a lesion, and even maybe predict which patients or animal will have a better chance of recovery than others, just seeing which one are actually showing signs of plasticity or these kind of things.
Mm-hmm. Yeah. To answer your question, I guess I'll split it in two parts. So I guess one thing is where the lesion is and how it affects, for instance, white matter tracts. So you might have a lesion that affects white matter tracts, and so potentially we could use imaging to identify if that person or animal will become better. So I think there are some preclinical studies that have been looking at that. We haven't looked at that specifically. Then the other thing is then to look at changes in the spare tracts that could potentially then also lead to recovery, right? So we have done some studies using a technique called neurofeedback in stroke patients, where we're using neurofeedback to induce alterations potentially or increase activity in the affected hemisphere or in the motor hemisphere, and we actually have found alterations in cortical spinal tract. And the talk will happen tomorrow at 8 a.m. by one of my students. So if you're interested in that, that'll be the neurofeedback session. So we have been able to actually identify alterations in white matter in stroke patients after intervention.
Yeah, another question.
Hello, this was really fascinating. I was wondering for the plasticity, if you have any ideas on response to, for example, direct electric stimulation, right? Can that play a role?
So you mean like TDCS?
Yeah, or for example, think DBS and the affected white matter tracts.
Yeah, I mean, like, I think it's possible. I mean, the animal work that uses things like optogenetics to stimulate specific neuronal populations, they do identify alterations in the white matter tracts that have been stimulated. So I think it's possible, but I don't know any studies that have tested for that. But yeah, I mean, the mechanism is there. I guess it depends how well the TDCS or whatever technique you're using activates the neurons and how specific it is.
Okay, right. Understand. No, I was thinking more about directly stimulating the white matter tracts, right?
Okay. Directly stimulating the white matter tracts.
Yeah.
You mean, yeah, okay.
Trying to somehow modulate the myelination, right?
Yeah, so the myelination is modulated by the fact that the neuron is active, right? So depending on the technique you're using, some techniques will basically lower the threshold of the membrane. I'm guessing TDCS, it probably works that way. So that could be a way of altering the neuron activity and modulating oligodendrocytes' ability to make myelin, for instance. But what matters here is that not the axon is active because of the neuron, right? So if you modulate the activity of the neuron, then you're indirectly modulating the oligodendrocyte response to it because they are sensitive to the activity of the neuron. Does that make sense?
Yes.
Okay.
If you could go to the microphone, please.
Hi there, thanks.
Hi.
Can you hear me? Okay.
Yes.
Thanks so much for your talk. That was a real treat to listen to. My question was regarding the diameter of the tracts. You mentioned that could possibly affect the plasticity of the cell. So I was just wondering if you could explain a little bit more how the diameter would have an impact on the plasticity of the cell.
Okay, so your question is, how does the diameter of the axon alter the physiology of the neuron or the axon itself? Okay, so the diameter of the axon, so larger diameters mean that the action potential actually travels faster. So areas where you have large caliber axons, you have faster conduction speed, basically. So any alteration in diameter will actually change the synchronization of the signals, for instance, which is actually the same thing that myelin is doing. It's just the brain has two mechanisms to change transmission speed. One is myelin, and the other one is axon caliber. And axon caliber is actually also a mechanism by which myelin is modulated. So myelination tends to occur in bigger axons. So in fact, most of the big axons above 0.5 micrometer diameter are all myelinated. Okay, so the little ones, the thin ones, are actually unmyelinated. So it's kind of like you have a fast route and a slow route in your brain. Does that answer your question?
Yes, thank you very much.
We have time for more questions.
Well, if I may slip a question in, please. You said that myelination is not the only factor governing this, but would you say it plays a major role, or would it only be like a 50-50? You know, if you had to give it a weight.
Play a major role in the MRI signal or in behavior?
In behavior, the plasticity.
Okay, in plasticity. I mean, I'm not forgetting we still have gray matter and synapses. So if we ignore all that, which obviously is the main mechanism by which the brain is plastic, so I'm not saying, oh, throw away all we know. No, I'm saying this is a complementary mechanism by which the brain is also plastic, okay? Now, if it is all myelin, I don't know. I think probably not. I think things like, in terms of plasticity, for instance, we know that nodes of Ranvier can change their structure, and this can happen really quickly. And this is also a much more energy efficient mechanism. So it could be that there has to be a threshold at some point that allow or drives myelination, but I don't think that that's all there is. I think there's other things that are happening in conjunction with myelination, like nodes of Ranvier altering their length, but also the caliber, the axon. I don't know if there's any evidence at which speed that can happen. That could be potentially also more, could be even just swelling, right? Faster, more energy efficient mechanism. But I think, myelin seems like something that is very energetic to do. We're having cells being, in one part cells being formed and differentiating. This doesn't happen that quickly. So the cell cycle from proliferation all the way to differentiation, depending on which brain area you're looking at, can take 10 days. Okay, so it's not that fast. Now, we know that some of these OPCs can differentiate directly, and that can be much faster. Like, for instance, in the optogenetic study, they start detecting this differentiation within 24 hours. It could be that some of the cells just went directly. They didn't go through the proliferation stage, so that would take 2-3 days. But the other aspect is that the pre-existing oligos can also potentially modulate the length, right? So this sounds that it could be something a bit faster, less energy efficient, more energy efficient, but there's actually less evidence for this occurring, which is something that we always assumed or saw in textbooks. But there's very little evidence, or there's controversial evidence for this to occur. What we know from most of what we know about myelination is it comes from zebrafish models, and we know that the retraction and formation of myelin sheets happens during a critical period and then becomes quite stable. You might have retraction, but they haven't seen formation of new myelin sheets that often after this critical period. That can be like 5 to 10 days. So there is some evidence that this can happen. There's at least 2 papers that have identified it, but they are a bit controversial because it's not something so easy to measure. It's not something that we currently have methods to look at.
Yes, please.
Thank you very much for your fascinating work. I was wondering, just after this question, I was wondering the same thing, but on MRI signal.
Mm-hmm.
And how much you, from your perspective and your experience, estimate that the changes of axon diameter or axon number can affect the MRI signal in comparison to the myelinization process?
Yeah. So the study that I showed at the end where they're showing the sensory deprivation, this was actually an auditory deprivation more specifically. So they show both things at the same time, right? So because in our previous studies, we were just looking at one of these things. So in that study, they see decreases in myelination and decreases in axon caliber. These 2 things have opposite effects in diffusion. We have never looked at caliber, so in our studies. Other people have looked at it, but it's still hard to estimate it's still hard to estimate how much of a myelin change in thickness, for instance, and how much of a diameter change you can have, and which one will have a bigger impact on our metrics, right? So how do you estimate that? Like if you have a change in thickness and then you have a change in diameter, how do you estimate that that will have X impact on FA, for instance? I think that's really hard to estimate because it's not just that. Then you have changes in extracellular space. You have a compact myelin versus uncompacted myelin. And again, if we're looking at this, we'd be looking at cross-sectional EM type images, and that just tells you a tiny little bit of the picture literally, right? So as I said, you can also have, alterations in myelin in certain areas of the axon and not in others. So I think it's just really a complex biological problem. So I'm not sure which one contributes to. I assume that all these things are happening at the same time, and sometimes we're able to detect it because, let's say, myelin wins versus axon caliber change, and so we detect it. But other times you might have, completely different things. And also don't forget astrocytes. Astrocytes have a huge contribution for things like the fusion. So if they're swelling, if they're changing their morphology, and I think a lot of the things that we're also detecting are astrocyte derived as well. I know I didn't answer your question, but I guess the answer is I don't know.
It's okay. Thank you very much.
We can take one more question. Any other question?
Get closer.
Oh, cool. Yeah, so my second question was about the level of myelination, if that depends on the task difficulty. Have you checked, like, if a easier task correlates with the level
Yeah, so that's a really good question. So if you remember in the first motor task that we had let's say a complex motor task. It's a fine motor movement task, which is more difficult. And we have just animals using their limb without any skill. So the movement itself is very different. So one is nice and smooth, and the other one is something like this. So in the MRI, we didn't detect anything, they're significantly different from each other, right? That doesn't mean that if we didn't have the learning group, maybe we would have detected something in that group compared to some other kind of passive control. So it's interesting because that's the motor task itself is actually something that rodents are not used to do, and that's why we went for it, because it's very novel. They're using their limb a completely different way. So that's why we went for that. But then when we look at other things, like there are less control, like for instance, the sensory experience tasks, we're also identifying alterations there, and other groups using other types of sensory enrichment, they're actually looking at the cells and they see alterations as well. They're looking at the oligodendrocyte and they see alterations. So it's a really interesting question, and I actually don't know what is the behavioral relevance or function here. Like I don't know what the threshold is. We also know that just activity alone can modulate myelination. So I don't know what the system is doing, or when is relevant and when is not relevant, when myelin is being driven and when it's not. But that's something that we're really interested in, and that's why we're using these knockouts, for instance. And we were looking at complex wheel running, but we also looked to see if there were significant differences, which we detected and replicated from other group. But if we put the animals on just normal motor stuff like rotarod, or which is just looking at if they have subtle deficits, they don't have anything, they're fine. So it's the interpretation of some researchers is that you need these new cells to acquire certain skills. But we also see that these cells and myelination is also recruited when you're just activating the brain, for instance. So it is a tricky question. There has to be an answer, but I don't know it yet.
Okay, that's fine. Thank you.
All right. So thank you very much for Cassandra for your wonderful talk.
Thank you, and thank you everyone for being here today. So my talk today is about various neuroimaging software that helps to unite structure and function in human MRI research. But you might notice that the word tutorial is in my talk, and this is a 25-minute talk. Sorry, is in my title, and this is a 25-minute talk. So I'd like to encourage you to think of this a little bit more like a software overview. I will include some demonstrations, but if you are interested in tutorials, I have gathered a bunch of tutorials for the software I'm going to talk about today in a GitHub page here on the screen. And in fact, I'm going to keep that GitHub page in the lower left corner of my slides for this entire talk, so you can find it easily. I highly encourage you to go to this GitHub page if you're interested in any of the software I talk about. The tools in this talk are of interest to neuroimaging research generally, but I study visual neuroscience as a domain, so I'm going to start by giving a very brief introduction to human vision. So shown here are the two inflated cortices of a human brain, but to make visualization a little easier, I'm going to just ignore the left hemisphere, and we'll focus on the right hemisphere. Now the gray trapezoid shown on the left represents a stimulus screen during a functional MRI experiment, and the white dot indicates the position at which the subject is pointing their eyes during the experiment. Here I've put a little bit of stimulus at the center of the screen near the subject's center of gaze. And I've colored the cortical surface by its bold response to the stimulus. Because we're looking only at the right hemisphere, and because the right hemisphere receives visual input only from the left side of the visual field, I've limited the stimulus to just the left side of the screen. Now, with the stimulus in the center of the screen, the cortical activation mostly occurs in the posterior cortex. However, if we sweep the stimulus out to the subject's periphery and back, the region of cortical activation will sweep towards the brain's anterior and back in a very clear pattern. If we were to take this pattern and visualize it as a single image, it would look something like this. Here, the stimulus is colored according to the distance from the subject's center of gaze, and the cortex is colored according to the part of the visual field where a stimulus produces a maximal response. So, for example, I don't seem to have a pointer, but for example, the posterior blue part of the cortex responds when the stimulus is in the blue region of the screen. This overall property is called the visual eccentricity. Additionally, we can measure the pattern of response when the stimulus sweeps in the orthogonal direction, that is, around the visual field. This stimulus induces a number of stripes of cortical activation that sweep simultaneously across cortex. Each independent stripe in this case represents a different visual area, and if we visualize these as a single property like the eccentricity, it looks like this. Here, cyan color corresponds to the upper visual field, and the reddish colors correspond to the lower visual field. This property is called the polar angle. An important feature of the organization of the visual cortex that we can see from these maps is something called the cortical magnification, which is actually a direct measurement of the relationship between the structure and the function of the brain. To explain this, let's look back at the eccentricity map briefly. Notice that the blue region on the cortex covers almost half of the visually active cortical surface, yet the blue region in the stimulus covers only a small fraction of the visual field. Inversely, the orange color that represents the periphery takes up a large amount of the visual field, but a relatively small amount of cortex. This is because the center of gaze is magnified relative to the periphery on the cortical surface. Calculating the cortical magnification requires a pretty good model of how the visual field is laid out on the complicated 3D cortical surface. That is, a model of the relationship between structure and function itself. And we will work up to that model in these slides. Before we get there, however, I will start out by pointing out that this measurement, cortical magnification, correlates well with visual acuity in individual subjects. And in fact, if you can calculate it very carefully, it correlates in individual points in the visual field with subjects' acuity at those individual points. Now, individual differences in the relationship between structure and function, such as those described by the cortical magnification, are one of the many reasons I find human vision so interesting. What you're seeing here is an animation of 3D brains that are morphing iteratively from one subject into the next and into the next, pausing briefly on each individual subject, for all the subjects in a dataset called the New York University or NYU Retinotopy dataset. The brains are colored according to each subject's polar angle response to functional MRI experiments. Simultaneously, the plot of the visual field at the bottom of the slide shows each subject's cortical magnification, where dark colors represent a low magnification and lighter colors represent a higher magnification. At first, this may just seem like kind of a fun just kind of a fun animation, but I also intend for it to demonstrate some important things about the relationship between structure and function in the visual cortex. First of all, it should be pretty clear that there is an enormous amount of variability in the shape and size of the visual cortex. In fact, if you look at the Cochrane sulcus, which usually I would point to at this point, but it's basically the great big groove around which all of the color that we're looking at is. If you look at the Cochrane sulcus, it's basically where the first visual area, V1, lives, and that visual area can vary by a factor of over 3.5 between subjects. Simultaneously, the entire cortical surface can only vary by a factor of about 1.5 between subjects. Despite this enormous structural variability, however, the general organization of the visual cortex is highly conserved, with the upper bank of this large sulcus in the back of the brain always representing the lower visual field, and the lower bank always representing the upper visual field. Unsurprisingly, these variations in structure lead to large variations in the cortical magnification as well. Making this animation that I just showed you requires a lot of software tools. For starters, someone had to run the actual MRI experiments and collect the raw data. And while this step is incredibly important for any neuroscience research, this talk is a software talk, so I will be focusing on the steps that follow the data collection. Once the data were collected, they had to be pre-processed, which in this case was performed using the tool fMRI Prep. And fMRI Prep itself calls a pre-processing tool called FreeSurfer, which extracts and calculates structural information about each subject. Once the data have been pre-processed, it was saved to a common file format called BIDS, which stands for the Brain Imaging Data Structure, then uploaded to a neuroscience dataset repository called OpenNeuro. Finally, I had to actually download the data, model it in order to calculate the cortical magnification, and render it as a bunch of movie frames. And for these steps, I used the repository access tool DataLad and my own Python library, NeuroPythy, for modeling and rendering the frames. These steps will basically form the outline for the remainder of my talk, during which I will step through these tools and provide an overview of each. And as a reminder, if you're interested in any of these tools, there are more resources and tools available at the GitHub page that I have at the lower left corner of these slides. We'll start by discussing some of the data processing tools used for this data set. The NYU Neuroanatomy dataset from which the animation we just saw was generated was pre-processed using the tool fMRI Prep, which I highly recommend due to its ability to standardize functional MRI pre-processing steps. This tool is one part of the Neuroimaging Preps community, or NEPREPs, and in fact, fMRI Prep is one of the main entry points for its software tools. fMRI Prep automates most of the structural and functional pre-processing of raw MR images. It handles details such as slice time correction, structure-function alignment, motion correction, resampling of functional data under structural spaces, and additionally it outputs the data in BIDS format, which we're going to talk about in just a second. fMRI Prep calls many other tools, including FreeSurfer and MRI-QC, which is a quality control tool for MR images. And these tools generate a number of reports in web page format so that researchers can easily see the quality of their data and pre-processing steps. The images shown here are examples of these quality control reports that are related to structural processing. Structural preprocessing itself is usually performed by FreeSurfer, or at least I usually perform it by FreeSurfer. FreeSurfer is a very mature tool that was developed back in the 90s and has gradually assumed a critical role in contemporary neuroimaging work. FreeSurfer performs a great number of usually tedious tasks in the structural preprocessing pipeline, such as brain alignment, removal of the skull, gray-white matter segmentation, surface tessellation, cortical inflation, and anatomical atlas alignment. The first many steps in the FreeSurfer pipeline involve identifying and describing the cortical surfaces. We don't have time in this talk to go into these steps in detail, but the basic idea is that FreeSurfer converts a 3D voxel-based image of the brain, such as the one you're seeing on the left, into 3D models of the brain's cortical surface, such as those shown on the right, using a series of finely tuned filters, heuristics, and algorithms. Another important utility performed by FreeSurfer is anatomical alignment. Now, in the animation I showed earlier, one of the key things that we would need to know in order to make that animation is how to morph one subject's brain into that of another. Doing this requires an understanding of how any given point on the first subject's cortical surface needs to move in order to land at the correct point on the second subject's cortical surface. In other words, we need the cortices to be aligned according not just to rotation and translation, but in terms of the folds at the cortex itself. The idea behind FreeSurfer's anatomical alignment is to normalize away the many differences in anatomy between subjects. For this process, we start by collecting structural MR scans, such as those shown here, from many subjects, these days typically more than 1,000 subjects. We then average their MR images together, voxel-wise, producing this image of a very smooth cortex. Now notice that the individual brains have a lot more folds and wrinkles in them, while the average brain captures roughly how an individual brain might be shaped. We can then extract a 3D model of the brain from this representation. And as an aside, we can actually make these 3D models from any structural MRI scan. And they are basically represented as triangles and vertices, just like modern 3D computer graphics. Next, we take this 3D model of an average hemisphere, which we sometimes call an atlas hemisphere, and is shown on the left, along with the model of an actual person's brain, shown on the right, and we can computationally inflate them. Inflation basically makes these hemispheres easier to work with because it turns them into spheres, and spheres are geometrically simpler. Once inflated, we might be able to see that there's some correspondence between the pattern of curvature indicated by the color map here, but the correspondence is not exact. So, for example, if we highlight the calcarine sulcus in the atlas hemisphere, that's the highlighted sulcus here, and we transfer that region over to the identical position on the individual hemisphere, we can see that it doesn't line up very well with the curvature of the individual. FreeSurfer corrects for this by treating the individual subject's cortical surface like a big rubber sheet that can be stretched and compressed but not torn or folded. By stretching and compressing the individual hemisphere's surface, FreeSurfer finds the optimal correspondence of curvature between the two hemispheres. This process looks a little like this. So as we can see, after this registration, the calcarine sulcus of the individual subject is well aligned to that of the atlas, and this is true for other sulci injuri as well. Overall, this creates a correspondence between the points on the subject's cortical surface and the surface of the atlas hemisphere. So if we then get another subject and align their hemisphere to the same atlas, we will have a correspondence of points between those two subjects, which allows us to morph one subject's brain into that of the other. This is also the method by which standard atlas models, such as the Wong 2015 probabilistic atlas of brain areas, are able to transfer an atlas over to an individual subject via this alignment. Once the data from visual experiments has been pre-processed, it would typically undergo a set of analyses in which we would solve what are called population receptive field models, or PRF models for each voxel. These PRF models result in the polar angle and eccentricity maps that I showed you earlier. However, this is all outside the scope of this talk, so we will instead assume that these PRF models have already been solved by vision experts, and we'll move along to data sharing. One of the most significant advancements, I think, in the sharing of neuroimaging data in recent years is the development of the brain imaging data structure, or BIDS. BIDS is a system for organizing both the raw data and the derived data from neuroimaging experiments such that they can easily be understood and interpreted by others. BIDS is an open self-governed standard that handles not just MRI data, but physiological, genetic, and behavioral data, and it works for many modalities, not just MRI, but also MEG, EEG, electrocorticography, PET, and microscopy. The image on the right here shows how a typical directory of machine-named DICOM files would be transformed into a set of files with standard formats for BIDS that includes metadata about the experiments. Such an organization is valuable in part because it enables both humans and computers to easily read and understand the data, thus enabling computational analyses to operate across many data sets. These features make BIDS critical to the sharing of data, and in fact, the website Open Neuro, which hosts the NYU Retinotopy dataset, that was used to make the animation earlier, is specifically designed for researchers to deposit and share BIDS datasets. Open Neuro is free to use as a repository of BIDS data, and datasets deposited there are freely available to others. The website gives each dataset a unique identifier that can be used to look up its data and metadata. The screenshot on the right of this slide actually shows the information page for the NYU Retinotopy dataset, which has the identifier DS003787 at Open Neuro. The dataset can be downloaded directly from this website. You can also get it using Amazon's S3 service or using tools like DataLad. This brings us to the final set of topics in my talk today, tools for accessing and manipulating the dataset. So we'll start with the DataLad tool. DataLad is a simple command line tool that's very similar to GET, but instead of interacting with code repositories, it interacts with data repositories. DataLad can be easily installed on any operating system by following instructions on its website, datalad.org. I'm now going to attempt a demo here using DataLad to download the NYU Retinotopy dataset. So hopefully here you're seeing a terminal window for my laptop. To start with, I'm going to go ahead and switch directories to a location on my laptop where I can store OpenNeuro datasets, which for me is just /data/openneuro.org. I will then run the command datalad clone, which is similar to the git command for cloning repositories from GitHub, and I will provide it with a GitHub repository which contains the metadata for the NYU dataset. You can see it ends with DS003787, which is the identifier. All OpenNeuro datasets can actually be downloaded in this basic way, and you'll notice that the download is rather fast. In fact, they're just finished. This is because only the BIDS metadata and directories have been downloaded while the data files themselves have not. We can now see that there is a directory for this dataset here, and if we traverse into it, we'll be able to see that it contains a directory for each subject along with standard BIDS metadata. For example, the participants.tsv file, which you should be able to see there, is a tab-separated value file that contains a list of basic information about participants, so we'll go ahead and just take a look at that. There you can see all the various participants, their ages, gender, etc. To make the animation we saw earlier, we would need to at least get data from the derivatives directory, which stores processed as opposed to raw data. So for example, we can see in the derivatives directory here that there is a directory for FreeSurfer as well as directories for example, ROIs that are hand-drawn, or there's a directory here called PRF analyze-vista, which actually contains the visual data we saw earlier. We'll go ahead and take a look at the FreeSurfer directory. And as you can see, there's this directory contains a directory for each subject, and each subject's FreeSurfer data is contained inside those directories. That said, the data files themselves haven't actually been downloaded yet because all we ran was datalad clone, so we need to go ahead and get these data. To do that, we issue a command datalad get, and we give it a list of directories or files that we want it to actually download. So here we'll go ahead and tell it to download the FreeSurfer directory, the ROIs directory, and the PRF analyze-vista directories, all of which are in the derivatives directory. And datalad will go ahead and begin downloading those diligently. And now you may notice that there's about 20 gigabytes to download, so this is going to take a while. We're not going to wait for it. You're just going to have to trust me that it'll finish. Once we've successfully downloaded the data in BIDS format, we just need a tool that can load, model, and render it. For this, we will look at my own library, NeuroPythy, which is a library of utilities for cortical surface analysis that is intended to serve as a sort of glue between structural utilities like FreeSurfer and more complex or nuanced analyses of cortical data. What you should be seeing here is a Jupyter Notebook window, which I will use to demonstrate some of what NeuroPythy is capable of. For starters, I'm going to go ahead and select that input cell, and I'm going to go ahead and import NeuroPythy with the alias ny. All right, so now I'm going to go ahead and define a few directories. So we'll start with the path for the data. So as a reminder, that was /data/openneuro.org /ds003787 for that data set. I'll then define the derivatives path, since we'll be loading data from the derivatives path, and that's basically in the data path /derivatives. I'll then define the FreeSurfer path, which is again in the derivatives path, so we'll put it in derivatives path /FreeSurfer. And then I'm going to define one more path, which is the PRF path. This is the path that contains the actual visual data. So this is in the derivatives directory /PRF analyze-vista. We saw that directory earlier as well. All right, now I'm just going to define a subject, and we'll just use subject 1, I think, for this example. And then we'll go ahead and define a session, and this just refers to the name of the MR session that was used in this data set to collect the visual data. Okay, so at this point, let's go ahead and ask FreeSurfer to load us. Actually, let's start by looking. Let's look at these paths just to make sure we got them right. So here we can see. Okay, so we can see all the subject directories here, including the one we're going to look at, the derivatives directory. Let's go ahead and look at the derivatives directory as well. Actually, the FreeSurfer directory. So we can see that we've got our FreeSurfer directory here as well. So these paths all look okay. So now at this point, let's go ahead and have NeuroPython load a free surfer subject. So for this, we just call the free surfer subject function, and we just pass it the path of the free surfer subjects directory. So for us, that's forgot my bracer there. So that's just the free surfer path followed by the subject's name. And this will go ahead and give us back an object. Let's take a look at it. This is an object called a subject object that knows where this subject is stored on disk. So at this point, let's go ahead and do a visualization. So we're going to use the function neuropython.cortexplot, which will plot the 3D surface, and we're going to pass it the subject's left hemisphere here. And we're going to ask it to plot the inflated cortical surface to start with. So when we run this, we'll get a 3D model here in the browser that you can actually interact with. This is the inflated surface for this subject, for the left hemisphere. Let's take a look at the white surface as well. So we'll just ask it to plot the white surface here. And that's a much more normal looking surface to me anyway. So here we have the white surface, so that seems to work fine. So at this point, let's go ahead and load some of the visual data. So we'll start by loading in the polar angle data. And for this, we're going to use the neuropython.load function, which knows how to load in most neuroimaging formats. We're going to pass it the PRF path we defined earlier, followed by the subject's directory, followed by a directory for the session. And then we're going to just give it the name of the polar angle file. NeuroPython knows how to read most of these files. Let's load the eccentricity as well. That's going to be pretty much the exact same line. We're going to load it from the PRF path slash subject slash session, and then it's just a different file name. And in addition, we'll load one more file, the coefficient of determination. And this basically is a file that tells us the confidence at any point on cortex that we have that it's visually active. So these values vary from 0 to 1, with 0 indicating that that part of cortex is not very active and 1 indicating it is. All right, so let's look at the polar angle here. And what you can see is we've just loaded in a vector of numbers in a NumPy array. And in fact, if we look at the shape of it, there's about 200,000 vertices, and that matches the number of vertices in the subject's left hemisphere, which we can see here. Now, if you're confused as to why these match, keep in mind, like I said earlier, these cortices are actually just like modern 3D graphics, they're just vertices and triangles. So if we zoom way in, you can see that this is actually composed of individual triangles and vertices here. There's a triangle, there's a triangle, there's a triangle. So this hemisphere actually has about 200,000 individual vertices, and we've just loaded in a polar angle value for each individual vertex. That's why they match. All right, so let's go ahead and plot some of these data. So once again, we're going to use the cortex plot function. Once again, we'll pass in the subject's left hemisphere. We're going to ask it to plot the white matter surface this time. And we're going to ask it to color it according to the polar angle we just read in. And just like with matplotlib, we need to give it a color map. There's a built-in polar angle color map. And we need to tell it the minimum and maximum value. So negative 180 to 180 degrees is the minimum and maximum for polar angle. And when we plot this, we should get a plot of the polar angle across cortex. Now, this doesn't look much like the animation we saw earlier because there's a whole bunch of noise over here. So I'm going to go ahead and add a mask. And for that, we're going to use the coefficient of determination that we loaded in. So we'll just put a mask here that says we only want to plot the color where the coefficient of determination is between 0.1 and 1, so where we have pretty reasonable confidence that the cortex is visually active. And that gives us a plot of the polar angle, much like we saw in the animation. Okay. So at this point in the talk, I've gone through just about everything in this animation earlier, or at least I haven't really showed you everything about it, but I've showed you a little about everything in the animation, with the exception of this cortical magnification plot at the bottom. And I mentioned that in order to make this cortical magnification plot, we need a very good model of how the visual field maps onto the cortical surface. So this is also performed by NeuroPython. Basically, we are going to use the exact same kind of method that we did with FreeSurfer, where FreeSurfer aligned two cortices based on curvature. But in this case, we're interested in a model of the visual field. So we can take a model of the visual field that's already been developed. Here I've just drawn it as a schematic. And we can basically just plop this down on this orthographic projection of the 3D cortical spheres. So that looks a little like this. I've drawn the model now using just lines so that you can see the data underneath it. What we're going to do is we're going to treat the cortical surface once again like a big rubber sheet, and we're going to stretch and compress the cortex. But this time, instead of aligning the curvature, we're going to align the underlying polar angle and eccentricity data to the model. Now, I'm only showing you I'm only showing you the eccentricity because I can only show you one at a time, but this actually works for both simultaneously. And that alignment looks a little bit like this. So that brings the underlying cortical surface into alignment with the model. And at that point, we can actually just ignore the data underneath and put a prediction based on the model. And we can relax the cortical surface. See if we can get that animation to work. There we go. We can relax the cortical surface back to its original shape, and we'll have a very nice prediction of how the visual field is laid out on the complicated 3D surface of the brain. And now if we want to calculate the cortical magnification, all we need to do is zoom in on a particular subject's cortex. We have a model of how the visual field is mapped onto the cortex. We can draw a little disc on the cortical surface. Here's one that's about 5 square millimeters in size. We can map that disc right into the visual field using our model, and it'll have a size there. Here this one is about 2 square degrees. And then the cortical magnification can be calculated just by doing some division. And we can do that across the whole visual field or across the whole cortical surface. All right, so I've gone over quite a bit today in this talk. If there was one thing I wanted you to remember from it, it's that the combination of structure and function in neuroimaging research leads to a lot of novel insights and measurements of the human brain. Additionally, we talked about structure and functional data processing tools such as fMRI-Prep and FreeSurfer, which have the power both to automate and to standardize a lot of the processing needed for these kinds of analyses. We talked about BIDS, which is a really critical standard for contemporary distribution of neuroimaging data. We talked about OpenNeuro, which is a free and open access data repository for neuroscience data. We talked about DataLab, which is a powerful tool for dataset management. And finally, we talked about NeuroPython, which is a useful open source Python library for manipulating and analyzing visual neuroimaging data. That concludes my talk. Thank you for your attention. I'd be happy to take any questions.
Now this talk is open to the questions. So if you have a question, please raise your hand, just go to the microphone. Yes.
Hello, hi. Thanks for the very nice presentation. I have a couple of questions. I'm interested to know, first, I think the pipeline that you showed is like super cool, super simple. Very impressed with DataLab also. You focused on retinotopy. I'm interested to know if this pipeline in the experience that you have was as simple to implement for maybe task-based fMRI data. Usually the data that you download, does it come with already projected contrast on the surface, etc.? And if not, that would be a second question. Does the library that you showed in the end, your library, handle volumic data, or like, how did you do the end? Thanks.
Yeah, and thank you. So let's see. First of all, the particular data set I analyzed already had the data projected to the surface, and however, that said, NeuroPython does handle volumetric data. It is really designed for surface-based data, but it does handle, in particular, interpolation from the cortical surface and back. So if you had a data set that was volumetric, so for example, even if you had a data set that was volumetric that had not been processed with FreeSurfer and you didn't even have cortical surface structures yet, you could just download the data set, run FreeSurfer on it, and then point NeuroPython at the FreeSurfer and the volumetric files and have it do the interpolation onto the cortical surface. That's all built into the data set, yeah. I think that I might have missed a piece of your question, but if so, just ask it again.
Thanks so much for your talk. I was just wondering, what's the advantage of using BIDs? Like, does it normalize the data better?
Yeah, so BIDS, it doesn't actually do anything. It's just a format standard. So the main value, I think, of BIDS, if nothing else, is that it allows you to put your data on openneuro.org for free. If your data isn't in BIDS format, you can't put it up there. And if you have a large data set, it can be really hard to find anywhere you can put it that is open access. Additionally, if you're ever going to send data to someone else, it's just, I think, polite to send it in BIDS, because if you don't, every lab has a cottage industry. Every lab has its own way of formatting its data and naming its data, and nobody ever likes to try to figure out some other lab's way of dealing with data, and BIDS solves all that problem. So I think that's the main advantage of it, really.
I see. And is there any storage capacity for that?
If OpenNeuro has a storage limit, I'm not aware of it. I think you just have to have it in BIDS format, and I think you're supposed to eventually publish a paper related to it, but I think those are the only requirements.
Okay, thank you.
Mm-hmm.
We still have time for questions.
Thank you for the talk. It was just a question about someone who does volumetric data analysis usually, and I wanted to know how easy it is to manipulate this kind of data on Python, because I'm used to just manipulate NumPy arrays, and I see vertices and things like that, and I'm like, is it hard? Is it easy to get into, or?
Oh man, that's a hard question, because it's been a long time since I've gotten into it. I've been into it for a long time, I should say. In my experience, there are some big advantages in how you think about the data that come from having it volumetrically organized, in that it's easy to think about slices and space in terms of voxels. It's a lot harder to think about space in terms of vertices, so I think that's the main challenge. In fact, if we were to look at that array of polar angle values that we read in, and we were to plot the individual vertices one at a time, you would see very quickly that there's no particular way that the vertices are organized on the surface. Vertex 1 and vertex 2 may be next to each other in the array of vertices, but they're often on opposite sides of the cortical surface. That's just kind of a reality of how cortical surface data works. But that said, that ends up being a relatively small problem in the long term. And libraries like NeuroPython include a lot of tools that make a lot of that much easier and much less problematic to deal with. On the flip side, I think that the advantages of the surface are very high, especially if you're interested in anything related to something like distance. Distances in the volume are very misleading, and distances on the cortical surface are much more meaningful, especially for gray matter.
Yeah, there's another question. Could you come over to the microphone?
Yes, sorry, it's me again.
No problem.
I'm not sure it's a question, it's much more of a fantasy, but since we have so much open source data at the moment, and a great deal of it is formatted in bits, sometimes I fantasize that we have this web-based viewer where we can look at the surface data that's been projected on the surface, but directly in the browser. Do you think, how far do you think we are from having this?
Yeah, all the technology for that is already available. It's just that no one's written it yet, as far as I know. So I think not very far. I think we're just kind of waiting for an enterprising lab or postdoc or grad student to write that software. And if one of you is interested in doing that in the audience, send me an email and I'd be happy to talk to you about it. But right now this kind of software is mostly in Python and a few other languages.
Any other questions from the floor to know? If not, so we will have to close and not so thank you very much. We still have some time for question, discussion. Maybe if you have any remaining question, maybe you can just go to the microphone to ask any of the speakers. cortex, it can be very plastic because even for we become adult, we can still run juggling, we can just run new skilled movement, but maybe it's not really generalizable to many other systems because other systems can be plastic. So do you have any kind of idea about how much degree of white matter plasticity is similar across different parts of the brain, or maybe motor is a particular place where you can just find plasticity easily?
So studies so far have looked at different systems. So the first system that people actually looked at was hippocampus, so fimbria and fornix areas. As you would expect, hippocampus is a very plastic structure, so people started looking there and they found evidence of that. Sensory areas, so in this case, because it's rodents, when I say sensory areas, I'm talking most specifically about the barrel cortex, which is the system that that senses the whiskers. So it's kind of the equivalent to the human visual system in rodents. I mean, maybe that's a stretch, but yeah, so rodents, you will use their whiskers to move around and to identify their environment, and you will use much less visual cues. I haven't seen, so yeah, of course, the visual system has also been used more recently. I've seen a couple of studies using particularly deprivation models. And auditory models as well. So it seems like, I don't know if the degree of plasticity is different because that's hard to compare, but it seems like there is myelin or white matter plasticity happening at multi-systems level.
Yeah, thank you very much. Yes, please.
Hi, my question is also for Cassandra in terms of the kind of language that we use to describe differences in white matter in the brain. So kind of often when you kind of read about, I don't know, differences people might see in white matter microstructure, people talk about reductions in integrity. So for example, a reduction in FA, that might represent kind of reduced integrity in white matter microstructure. But, I think I've been reading recently about actually maybe that's not an accurate representation of what's going on kind of at a physiological level, especially in terms of crossing fibers, but now also in terms of, the differences you see in, the kind of diameter in axons and other kind of cellular functions. So I'm just wondering, do you guys have any thoughts on the language that we use to describe these differences in white matter when we're reporting them in our research?
Yeah, so people tend to use the word microstructure when they're talking about healthy conditions, and then they also use integrity for maybe more non-healthy conditions. Both are umbrella terms, so people use them interchangeably. So I mean, language is really important, but in this case, to be honest, I mean, because we don't know what we are measuring, I would say I'd like to use microstructure, so alterations in microstructure in white matter. That won't tell you anything much because, again, we don't know what is changing exactly, right? Integrity, I've seen it more when people are referring to pathological conditions like MS or stroke, where integrity is affected. If people might associate integrity maybe with axonal damage, but again, we don't know what's measuring, so it can be anything. We could be more precise, but because the technique itself is not really precise, I'm not sure which umbrella term is best, to be honest. But yeah, I think you can always explain the caveat in your research paper that this is an unspecific measure. And so it can be modulated by all these different things, and give a couple of examples just to make sure that people understand you're not saying this is myelin or these are axons.
Great, thank you.
Any other questions from the floor to any of the speakers?
Yeah, if you go up to the microphone.
Hi there. So this is just a general diffusion weighted MRI technique. So usually in diseases, we see the FA increases and, I think the FA decreases and the MD increases in diseases. So what if we see the opposite in a disease? So why do you think we might see the opposite, like a decrease in MD and increase in FA?
Okay, so specifically MD. MD is just a measure of how restricted the water is regardless of direction, right? So for instance, in any tissue really, if you have more water or if you have tissue damage, or if basically you have no restriction due to changes in for instance, fibers or the density of fibers, and I'm talking even the cortex because you can use that MD in the cortex, then you will have increase in MD, right? So thinking of pathological conditions where you can have the opposite, potentially in areas of crossing fibers, where you have maybe one of the fibers being degenerated, then you can have that effect. And then an FA would have the opposite effect, obviously. Does that answer your question?
Yeah, sure. Thank you.
Could you come closer to the microphone, or maybe it's not switched on?
Can you, oh, yeah, there we go. So I guess this is a little bit related to the last question. Have any of you, maybe those more familiar with white matter, heard of the eye condition of, it's actually called myelinated nerve fiber, where there's too much myelin on the nerve fibers in the eye, yeah? I'm just curious if you have any ideas of why too much myelin would cause visual problems. Generally, I mean, I don't work at this level, so I'm not as familiar with the research, but generally I thought more myelin would help the signal. I'm not sure whether it physically blocks the light going into the retina or if it somehow disrupts the signal along its path.
No, I don't know that condition specifically, so I don't know if it is blocking the light coming through the retina, but myelin in general, it will speed up transmission, right? So if you have too much myelin or too little myelin or an alteration in myelin, you're changing the circuit, right? So you might basically cause desynchronization of the signal instead of synchronization of the signal. So if two signals have to arrive to a particular brain area at the same time, and now you're altering myelination on one side and not the other, then they're not arriving at the same time anymore. So these things have to be fine-tuned. How the brain does this specifically when it comes to myelin, I don't know. It's a very interesting question that I'm interested in. But you can imagine that if you have too much myelin, that can be a problem if now the signals are no longer synchronized.
Okay, thank you.
You're welcome.
Any other question? Maybe stay for some comment. Yes, please.
Is it still? Oh, okay, perfect. Throughout the different presentations, there was emphasis on the fact that structure and function are really related in different manners. But from time to time, we could see also that there was variability between where certain functions are situated compared to some certain gyri or sulci. And also, when we do registration of brains to a template, usually we only use, well, most of the time we only use the structure of the brain. And I was wondering, because I've seen a few articles doing functional registration, but do you think this is something that should be more widespread and should become a new standard, or we should still keep doing registration mostly on the structure of the brain?
Yeah, I think I can answer. So I guess just for some context, a good example of uniting structure and function in the registration process is the Human Connectome Project. They collect a bunch of data, including resting state data, and use that in their their generation of what's called the FSLR template, which is similar to the FreeSurfer template, but obviously uses this other functional data as well. My intuition, having used it and FreeSurfer's registration, which is structural only, is that they're pretty similar for most research, but they're not identical, and that the FSLR template is better for certain things. I can't comment as to whether it's always better. I just know it's better for some things. But my intuition about the question of whether we should continue using structure or structure and function is that you use them for different things. If you want to isolate structural variability, you want to align using FreeSurfer. If you want to get maximum alignment between subjects, you would probably want to align using both function and structure.
So, you know, I'm a post-mortem person, so I'm going to ask a follow-up question to this. What about the single subject analysis? I mean, in the past, from what I know, functional imaging studies concentrated on creating the average, and that was what they said. This is the area involved in whatever function. And now I think the trend is moving more towards doing the actual analysis in the individual brain space. So that gives you a better correlation between the activation and the anatomy of that subject. Because the point that you were making concerning there's a different location of the activation focus with relation to a macroanatomical landmark. It was so funny when you were presenting your video. I thought, oh, look at that, the brain seems to be changing more in the occipital pole than in the frontal pole. I wonder if that is something like generalizable, or is it just because you're presenting data from the visual system, so of course the color changes are happening in the visual system?
Yes, that's a great question, and actually, I haven't calculated anything that would say, this part of cortex is the most changing between subjects, but my intuition is actually that the occipital cortex is probably the most changing between subjects, or the most different between subjects. And the reason for that is largely that when we look at the occipital cortex, first of all, in that animation, you were seeing real differences. The occipital cortex does change a lot more between subjects, and what we found is that, I think I said in the talk, we can have variability of almost 4 times in terms of magnitude of just the size of V1, the first visual area, whereas cortex only varies by maybe 1.5 times across subjects. So that variability in the occipital cortex has to be made up by stability somewhere else. And it's possible that auditory cortex and sensory motor cortex, for example, are equally variable, or that there are some parts of frontal cortex that are even more variable. But my intuition, at least, is that occipital cortex is one of the most variable. Yeah.
Fascinating. In macaque monkeys, the highest degree of variability, this structural variability, is also found in the occipital lobe. Funny, I wouldn't have thought that. If you think that when you're moving from non-human primates to primates, actually the greatest degree of change happened in the prefrontal cortex. And then it's sort of like, it was surprising for me to see that actually then within humans, there's a greater degree of individual variability in the occipital.
Well, and something that actually deepens the mystery a little bit is that most people's intuition, if you just ask them, is that the first visual area, like the first stop for information from the eyes on cortex, would be less variable than the next one, and that as you go higher up the visual hierarchy, up to like face areas, for example, you would have more variability. But we actually find the opposite. We find that the most variability is in primary visual cortex. V2 and V3, which are around primary visual cortex, have lower variability, and as you go higher, it seems to get even smaller. So it almost seems like the source of the variability might be the visual world that you experience, but I'm speculating a bit here.
Right, because you haven't done, or do you know of studies that have done this kind of analysis, but for the auditory or the sensory system? Like, would it be something specific for humans? Because as Cassandra said, you know, we humans, we use our visual system to well, and our hands, you know, but we're visual beings. Whereas, for example, rodents would use their whiskers to explore the environment. So in rodents, you'd expect this kind of variability in the somatosensory system, and because we are visual, you have it in the visual system.
Yeah, absolutely. I mean, visual cortex is already a little over 25% of cortex, so it's the biggest sort of, if we think of occipital cortex as like one unit of cortex, it's probably the biggest single unit. So that it makes sense that the most variability would probably also be found there for humans.
Hi, can you hear me? Yep. Well, thanks everybody for the talks. They were all super interesting and fascinating. This is just a very small follow-up comment on what Noah Benson was just commenting on the variability in V1 and downstream areas. And I was wondering, I'm not aware of those studies, but my question relates to it's clear that I also encountered that the variability in the size of V1 is like really remarkably large. I know that that also happens in macaques. Now my question is, I've also encountered quite a bit of variability further downstream too. So my question is if you have you can clarify under what kind of criteria you're making this decision that further downstream is actually more similar compared to kind of earlier areas.
Yeah, so we did a study of the Human Connectome project, which had about 200 subjects who participated in visual mapping experiments. And we actually hand drew boundaries for V1, V2, V3, and are on the process of drawing them for higher visual areas. And we just calculated the coefficient of variation of the size across areas, and that's very clearly decreasing as you go up the hierarchy, or as you go downstream.
Okay, that's interesting. But there might be other kind of criteria, for example, under which, for example, you can say one subject does not have a specific kind of selective area, and then you might think, okay, then it cannot be more different than that because everybody has V1. So it's interesting that maybe different criteria can kind of describe different that kind of variability.
Sure. For example, you might have downstream areas whose location are more variable, but whose size is very static, for example. Yeah, that's a good question. That's something we haven't looked at particularly. Yeah.
Cool. Thank you.
One thing I can add, I think there is old paper in 1997 by Andrews and they published, I think it's Jamie was, I think they're doing some post-mortem section to identify V1 based on the analysis, right? Then they also found a very large variability in V1. It's about maybe small person, large person, with 3 times differences. So I think it's not the variability more very specific to real image data as well. Any other questions, comments? Yes.
Again, a follow-up to what variability in lower versus high regions. Do you think this could be related to the task that's at hand? Like if you're maybe doing a grading discrimination or something, maybe that depends more on your lower level, like visual skill that's learned over the course of life, whereas if you have like a naturalistic movie or something like that, which depends less on the skill, but more on the subjective perception, you might see more variability in higher level regions. Would that make sense?
Yeah, so that's a good point, and this is something we absolutely need to look into. It's possible that the stimulus, which is really, honestly, our stimulus is mostly designed for V1. Part of that is that there's only a vague notion of what higher order visual areas are doing. We have a sense of what V2 and V3 are doing, but we don't have a model of it. That said, all of these visual areas do have what we call retinotopic maps, where the whole visual field gets represented in the area. And so we can use these stimuli to draw the visual area boundaries that pretty much tile cortex. However, I totally agree that there's, we've seen in some areas, such as the IPS regions, that the task, whether you have a task that requires you to attend to the stimulus or attend to the fixation dot, will change the appearance of the maps. So we know some of that happens in higher order areas. We just don't have a very good sense of how much, and I think that in order to answer a lot of those questions, we need better models of what higher visual areas are doing. Yeah.
Thank you.
Mm-hmm.
Hello? So in the beginning of the session, you spoke about how people used to argue over drawing boundaries between brain areas and such, and you just mentioned how you're doing this, you're drawing boundaries between brain areas. And I was just wondering, how do you kind of ensure that you're getting the right boundaries? Do you have to have, 10 people doing it and then take the average, or do you do one and have someone else check it, or how does that work?
Yeah, so we've been drawing boundaries for V1, V2, and V3, and we're working on higher order ones. Interestingly enough, in V1, V2, and V3, human raters agree with each other very well, and the main reason for that is because the boundaries are quite clear, typically, in V1, V2, and V3. That said, even as we were working on those boundaries, we realized that there were features in the V3 map that weren't really explained by the standard model of the retinotopic organization in early visual cortex, and so none of this is all that easy or clear. And in V1, there's been a lot of work done on V1. We have drawn it in thousands of subjects, really, across all the research that's been done, and we have a pretty good idea of where it is and what it does, and or approximately what it does and how big it is and that kind of thing. But you get very far into the visual hierarchy, and those, you can talk to experts and they'll be able to draw the maps pretty reliably, but they won't necessarily agree with other experts all the time. They'll for certain areas we're very good at it, for other areas we're just not. There's a lot of idiosyncrasy in the maps in like lateral occipital cortex, for example, and I would basically just say that this is an active area of research, and we're still trying to formalize how these maps are arranged and whether there are real individual differences between subjects in their sort of topology. Yeah. Yes.